The overall goal of the following experiment is to investigate how the looping dynamics of double-stranded DNA are affected by the intrinsic shape of DNA without the use of DNA binding proteins. This is achieved by incorporating dye molecules into double-stranded DNA fragments of different curvatures, so that DNA looping can be monitored by Fluorescence Resonance Energy Transfer, or FRET. The reversible looping and unlooping events from the single DNA molecules can then be observed by total internal reflection microscopy. The looping rate of the DNA can then be extrapolated from the time trajectories of each single molecule fluorescence. Ultimately, the looping probability of the DNA in relation to the intrinsic shape of the fragment can be measured.
The main advantage of this DNA looping assay is that it doesn't rely on a DNA binding protein, which might affect the looping kinetics of the DNA.
The simple protocol is about using a technique called Fluorescence Resonance Energy Transfer and PCR-based DNA synthesis to measure the looping probability of DNA.
Begin by designing globally curved DNAs by repeating a 10-mer sequence. For example, this representative sequence is a 186 base pair curved DNA, where X is a random extra base, and the sequences flanking the repeating 10-mer sequence are adapter sequences. Next, perform PCR with primers 1 and 2, with FRET donor Psi3 labeling of primer 2 at the 5' end. Then, perform PCR with primers 3 and